interaction and the host pathogens relationship. And Dr. Nahamani is actively involved, particularly in the the important projects which is related to viral diseases in soybean and other crops, and especially soybean seed diseases and the seed transmission mechanisms. And also she's working on tomato also. And she's also contributed to training and capacity building programs in seed science technology. And she has published several research papers in international reputed journals and contributing significantly to the plant virology and seed health research. And she's also got a lot of research awards. And with these brief introductions, I warmly welcome Dr. Nahamani Chandra and request you to kindly deliver your lecture. Thank you.
Thank you, Dr. Selva Kumar. I think I am audible to each and everyone. So today we will discuss about the topic advanced plant disease management techniques of vegetable crops under protected cultivation. So before discussing the advanced plant disease management techniques, first of all, we have to understand some basic things, why we have to go for protected cultivation. Because if we want to maintain the production throughout the year, and some crops which are very sensible to the fluctuating temperatures and humidity conditions, and so that we will go for the protected cultivation where everything we can control, like temperature and humidity. So that is the basic idea of the protected cultivation mainly, and we can manage the inflow and outflow of the insect vectors, which can act as a vectors for the many plant diseases, and we can maintain the production throughout the year. So that greenhouse and polyhouse net house protected cultivation means it involves all these things, so which will helps us to year-round production. And because of high humidity, because in this protected cultivation, that is which is covered structure, there used to be sometimes maintenance of high humidity and dense canopy if you are not following proper spacing. So this dense canopy, they will favor the pathogens. So in such cases, disease management is the critical issue for getting successful crop. Because in here, under protected cultivations, we will maintain the temperature from 18 to 30 degrees centigrade and relative humidity 70 to 90%. And because of poor airflow, if any humidity maintains there, then major diseases we will observe, like fungi and bacteria and viral diseases. So mainly first we have to understand why these diseases develop. As you all know about the plant disease triangle, like host, if you are taking the susceptible cultivars, which is susceptible to some pathogens, so that will favor the disease development very fast. And particular cultivars within the same crop, if we will take the tomato, we are having lot of hybrids. In that hybrids are some cultivars, like if we'll take suppose in Pusa, Ruby, Punjab, Chwara, they are very highly susceptible to tomato leaf curl New viruses. Whereas some other varieties are there, like Pusa presence fruit, they are very resistant to this tomato leaf curl New virus. So that is the based on susceptible nature of the cultivar, and pathogen, that is virulence nature of the pathogen, like fungi, bacteria, viruses, and nematodes. And what is the virulence? There are different strains of the pathogens used to be there, like a high virulent strain and a low virulent strain. If it is a high virulent strain, and then it will cause major disease. If it is a low virulent strain, then it will cause less damage to the crop. At the same time, favorable environmental conditions. If high humidity is there under the protected conditions, like soil fertility and what type of soil we are using under protected conditions, because it is very difficult to replace the soil also under protected conditions. So if we are using the same soil type, then there is a high build of fusarium diseases, especially in case of these protected conditions. So based on all these conditions, development of the disease, it will depend. And what are the major diseases can we observe under protected cultivations, like fungal, powdery mildew, downy mildew, and botrytis and fusarium wilt? These are the major fungal diseases we can observe in tomato, capsicum, and cucurbits. And in case of bacterial wilt disease, that is Ralstonia wilt used to be there, and leaf spot disease. And in case of viral diseases, especially tomato and if capsicum, we will take, and even in cucurbits also, if we will take, there is a problem of tomato yellow leaf curl diseases. The tomato yellow leaf curl diseases, that is a problem in cucurbits also. That disease is caused by majorly nearly 5 to 6 different variants, like New Delhi virus, Palampur virus, Joypur virus, like that different variants are there in the tomato yellow leaf curl disease. And another major pathogen in we can observe in protected cultivation is the cucumber mosaic virus and tomato spotted wilt virus. Tomato spotted wilt virus is not a problem under Indian conditions, but in other countries under protected cultivation, this is the major problem. In case of nematodes, we generally observe the meloidegon, that is root knot nematode. These are the major diseases. Here you can see some of the pictures how the symptoms will appear after the infection. In if you will take in the first row, you can see the powdery mildew infection, where initially the spots will appear and later it can cover the entire leaf. So what it will do, it will reduce the photosynthetic efficiency of the plants. Automatically, if the photosynthetic efficiency will be reduced, then that fruit formation also will be affected. So then later that yield, total yield will be reduced. And in case of second row, if you will see, there is a botrytis infection where in case of botrytis infected fruits, there is a rotting. You can see how the tomato fruit after infection of botrytis, it is looking like. And here in capsicum also you can see. And this is a third row, you can see the fusarium infection where fusarium wilt, because this is the mainly fusarium is the both the soil and seed-borne pathogen, but generally fusarium wilt occurs due to inhibition of the soil with the fusarium. So these are the different type of symptoms which can reduce the market value of the product, because ultimately we need the higher yields and good market value for the product what we are producing under protected cultivation. So because of these diseases, ultimately our yield will be reduced and market value, because the seed also we will get very less seed, and fruit quality also ultimately used to be reduced. So in case of this tomato and capsicum, we will observe powdery mildew, grey mold, which is caused by botrytis and fusarium wilt and leaf curl viruses. And in case of cucurbits, mainly we observe the downy mildew and the powdery mildew, that is Podospira and Spirotheca. And the root rots like Pythium and Phytophthora, and the gummy stem blight is there. Gummy stem blight in case of cucurbits is caused by Didymella. And viral diseases, cucumber mosaic virus and Juchini yellow mosaic virus. These are the major issues. Nowadays, Juchini yellow mosaic virus is the major issue under protected cultivations, as a lot of private companies, they are facing a lot of problem because of this virus. And due to seed transmission nature of this virus also, they are facing a lot of problem. And if you will take the leafy vegetables, major issues are the damping off, which is caused by Pythium and Rhizoctonia, and the sclerotinia and bacterial leaf spots. Here in the first picture, you can see how the leaf quality, like which we will use it as a salad. So how the leaf quality will be affected due to because of these bacterial leaf spots and poor seedlings, which affect the whole crop. And here you can see the sclerotinia rot in the second picture. You can see how it will start from the bottom and it will infect the total plant. So total yield used to be reduced, because in leafy vegetables, leaf ultimately we will use for the consumption purpose. And if you will take the integrated disease management, before going to the advanced techniques, we will see what are the different management techniques we can follow. First one is the this is the basic criteria we have to follow for managing any disease, because as a single technique will not give any effective management of the plant disease. So from the starting onwards, we have to follow cultural practices and host resistance and grafting mechanisms, biological control, chemical control, and environmental manipulations and monitoring and diagnostics. So in diagnostics also, we will discuss about some advanced diagnostic methods. If we will see the cultural practices like use of disease-free seedlings. So before choosing the seedlings for transplanting, so we should ensure that they are free from any pathogen. That is, we have to test for the pathogen that there is no pathogen in that seedlings, like tomato seedlings and capsicum. What we will do transplanting in the greenhouses. And the nurse maintenance of the hygienic conditions under the nursery, and use of sterilized growing media, because to grow these seedlings from the seeds, we will use the pot mixtures, like whatever potting mixture we are using, vermiculite or soil, right? That should be sterilized to, because we should not reuse the same soil mixture repeatedly, to avoid the this burn up soil borne pathogens, especially in case of fusarium. Then disinfection of trays and tools, and then we have to follow the proper spacing which is recommended for the crops, instead of dense sowing and dense transplanting, like, and pruning and ventilation is the main criteria we have to see to avoid this pathogen accumulations. And drip irrigation and sanitation, we should not allow the sprinkler irrigation in case of these protected conditions, because if the bacterial pathogen, if it will infect the single crop because of this rain splashes, it will disperse to the other crops also. So in case of protected cultivation, better to go for the drip irrigations instead of the sprinkler irrigations. And the sanitation practices and crop rotation also, instead of going for the single crop throughout the, like, if we are using 3 seasons, 2 seasons, so we should not go for the same crop, we should rotate. Like if we are using cucurbits one time, then go for the solanaceous crops next time. So crop rotation should be practiced. For disinfection purpose, we can use a sodium hypochlorite solutions or mercury chloride as per recommended practices. First, we should disinfect the whatever equipments we are using under the protected conditions. Then coming to the host resistance and grafting, that host resistance is the best available technique to manage the disease, beside keeping all these fungicide applications, all these things. If we are having the resistant cultivars, our best option is to go for the resistant cultivars. Like if we will see, already I told that in case of tomato leaf curl New Delhi viruses, which is a major problem in tomato, which lead to no fruit formation. If once the plant is infected, then automatically total fruit yield used to be reduced. So we are having like Pusa tomato hybrid 6 and Pusa Rakshit and Pusa Prasanskrit. These are the different types of varieties, like Pusa Prasanskrit, which is a cherry tomato, and the Pusa tomato hybrid 6 is a normal tomato. So these are resistant to, at least they can tolerate the tomato leaf curl New Delhi virus diseases. So we can go for the resistant cultivars. And grafting technique, recently they are following, but grafting is not yet successful, like grafting of the brinjal and tomato plants to impart the resistance to especially this fusarium. So this is not yet came into existence because it is under trial conditions. So then coming to the biological control, because nowadays government is also banning the most of the chemicals. We are having other alternatives like biological control, like pseudomonas and bacillus and trichoderma. These will be useful. Trichoderma especially useful for soil-borne pathogens like epithium, rhizoctinia, and fusarium. Whereas Pseudomonas fluorescens and bacillus we can use against bacterial and fungal diseases. And if you want to improve the plant growth, then we can go for arbuscular mycorrhizal fungi and mycorrhiza, which enhances resistance. How they will enhance the resistance means, if we are applying the PGPR, like plant growth promoting growth regulators, they, what they will do, they will enter into the plant and they will automatically in the surrounding nature of the roots, they will increase the beneficial microorganisms, so that some resistance, like induced systemic resistance and systemic acquired resistance, both will activate in the plants. So because of this resistance, plants get some impart the acquired resistance to face the pathogens. Because of this resistance, the influence of the that pathogen used to be less on the plants. So in such a way, PGPR can enhance the plant growth, and they will activate the resistant mechanism. So thereby they reduce the this disease influence. Another one is, only problem is that in case of biocontrol agents like trichoderma, if you will take, what is the shelf life of that trichoderma cards what we are using? Like 6 months generally we will say that shelf life. And what are the, because after receiving ourselves whether it is a viable trichoderma or non-viable trichoderma. That is a major issue for using these biological controls, because under which storage conditions the companies are maintaining these biological agents, whether they are viable or, because only viable bacterial they can enhance the plant growth and they can enhance the resistance mechanism. That is the only disadvantage we are having in biological control for using. And if you will take the chemical control, we are having a different type of chemicals. So based on the infection pathogen we are getting, then we have to go for these chemical controls, like strobilurins are there, mancozeb generally we will use for the downy mildew, that is lower fungi, and trizoles are there, which are broad spectrum control, like different leaf spot pathogens, and powdery mildew we can use. And at the same time, we should not repeatedly, like 4 sprays, 3 sprays, we should not use the same chemical. So instead of that one, we can go for like contact fungicides and systemic fungicides, then broad spectrum fungicides, which will act on the broad different pathogens. So better to go for rotation here also, like a crop rotation, how we are doing here also. One time if you are using contact fungicides, then next time better to go for the systemic fungicides. And before going seed treatment also we can follow. Then again bactericides, like based bactericides, COC is there, copper oxychlorides, which can manage the bacterial diseases also. So these are the different chemical management practices. We cannot say that it is a control technique, just we can say that pathogen management, because we cannot completely control the diseases. We can reduce the pathogen density and pathogen inoculum. Then most one is the physical environment manipulation, which is we are having in our hand under protected cultivations, because if we control the humidity like up to the which is not suitable for the pathogen growth, and temperature maintenance, like, and if you are providing good ventilation, these things are manipulated, like we can manage these things. And use of insect proof nets and good drainage, because we should not we are using two type of entries in the protected cultivations. So because of that, we can prevent the entry of the insect vectors inside the glass house, because especially in case of tomato and this capsicum, this tomato leaf curl noduli viruses, like if single white fly which is infected, which is already infected with this tomato leaf curl diseases, then it will spread completely to the other crops. It is very difficult to manage, because viruses we don't have any management technique. So only we have to manage the insect vectors. Even after managing insect vectors, we cannot ensure that these plants are completely away from the disease, because of this tomato leaf curl disease, total crop used to fail. Another one is the like ultraviolet treatments we can use, that is UVC within the range of 100 to 280 nanometer range. They will also helps in reducing the like pathogen inoculum on plant surface, and ventilation and evaporative cooling. These are the environment manipulation techniques we can follow to reduce the pathogen inoculum. And disease diagnosis and monitoring, like regular scouting, like if you are using advanced techniques and like sensor based devices, I will tell in the coming slides. And ELISA and PCR for confirmation. ELISA is the technique where we can screen the infected seedlings. So I will explain the technique, how we can use this technique for screening mechanism. And sensors and AI for early warning. These AI systems like artificial intelligence. So here you can see the detection of multiple viruses. So multiplex PCR is there, one of the technique, where we can detect multiple viruses in a single reaction instead of going for single PCR based detection. We will see all these techniques in a different presentation. I will explain after this presentation. So what are the major challenges we are facing in disease management is because of high disease pressure in enclosed systems. Once the pathogens start developing, then automatically spreading of the pathogen is very easy under protected cultivations. It is not like open field conditions. And even if you are using regularly the fungicides, so there is a resistance development in that fungal strains, and it is a high cost and limited technical knowledge. We don't have that much technical knowledge, especially in case of viral management. Then climate change impact is under open field conditions. This is not for this protected cultivations. And we are having regulations on chemical pesticides, like already I told, seed treatment. In some European countries, seed treatment is banned with thiram and thiram chemical. But here we are using for the seed treatment. So if we will see the some advanced techniques like artificial intelligence based disease detection and rapid biosensors and diagnostics, and like smart greenhouses and Internet of Things. And recent technique for recently we are using the CRISPR based disease management strategies, like targeting the host gene, which is the target for plant viruses. So we can silence a single nucleotide through the CRISPR-Cas system, so that the target will not be available for the plant pathogens, so that we can impart the resistance to the plants. But this is up to not full potential. It is just under the experimental trials, a CRISPR based technique for the pathogen management. And the microbiome manipulation, like if we are engineering our microbial communities, like nowadays people are in the process of developing super biofertilizers like agitobacter, all these things. If we will improve the biofertilizer efficiency of that bacterium, then microbial communities we can increase in the soil, so that they will impart more resistance to the plants. So if we will see the AI based disease detection, in AI based disease detection, first what we have to do, we have to collect the lot of images around, like people used to collect 10,000 images, 10,000 to 15,000 images. With the help of either we can take the smartphones, or drones, or CCTV, or sensors which used to be fixed in the greenhouse glass houses. We have to take first images of the both healthy plants and diseased plants at a different stages, like seedling stage, then after little bit maturation, then at plant growth stage, and before flower initiation stage, during flower initiation, like different stages. We have to take from the different angles, different photographs, like from back side, from front side, like that. We have to take different images. Then after images, then the second step is the data pre-processing, like for improving the any model performance. Then we have to make the images very clean, and we have to remove that noise, like what is the background of that images, and what is the size of images, that all sizes should be uniform. And we have to normalize the what is the brightness and the contrast of that images. Then we have to label those images, whether it is a diseased one, whether it is a healthy one. We have to label those images with the names. Then we have to curate this data, like whatever images we are getting. Then we have to mark the specific infected regions with the, we are having different tools like the label image and the CVAT. These are available through online. We can use these CVAT and label image data annotation tools. Then by using this CVAT, then we can create the boundary boxes or segmentation marks. Like if in entire leaf, if particular disease is happening in the middle of the leaf, that area we can mark. Then we can create the bounding boxes for that disease, like segmentation, with the help of this CVAT or label image, this data annotation tools. After creating all those things, then we have to choose the models. Generally, people are now using CNN networks and deep learning models, like conventional neural networks and these models, because whatever curated data we are having, then we have to give to these models, whether whatever data we are collected, whether it is accurately processed or not. like how much accuracy we need, and which device we are using for taking the photographs. Like mobile, we are using our server based images, then we have to give to these models, either CNN or deep learning models. Then we have to train to that model, like that these are disease based images and these are healthy images. Like that we have to give training to that models, like feed label data into that model. And then we have to see that whether color change has to be there, and leaf texture and spot patterns. And we have to training like multiple iterations, like optimization of accuracy, how much accuracy is there about that image, like disease if we will take and healthy, then how much accuracy based on that color variation also we have to see. Then finally we have to do that model validation and testing, whether that model like the CNN and this deep learning models, whether they are accurately processing our images or not. We have to test that model, whether that model is exactly suitable for our purpose. Then we have to evaluate the accuracy of the model and precision, because how we can evaluate means next time again for taking more number of images and again subjecting to the same models. Then after checking the accuracy, precision and recall, then we have to see that whether it is this based on these images, these models are able to detect the or diagnose the pathogen under the protected conditions. So integration of these models into the mobile apps or greenhouse monitoring system and IoT, that is Internet of Things devices. So we, after checking the accuracy, we will integrate these models like CNN model or deep learning models into these monitoring systems. So based on, then after integration of these monitoring systems, then if any high humidity is available under the protected condition, these models will helps to predict that this in particular area high humidity used to be there. So because of that high humidity, any of these pathogen may infect that plants. Like that, some signals used to be given. So then these models helps in disease detection, and classifies helps in whether because of high humidity fungal disease, and whether dense canopy which disease we can get, and bacterial disease. And it will also give confidence level. So based on that, we can recommend to the farmer that what are the different control measures they have to follow, and what is the suitable fungicide based on fungus which infect the plant. We can suggest which fungicide he has to go, and what are the different cultural practices he has to follow. And again again we have to give like this same process we have to repeat for giving different diseases data to these models. So again we have to collect new data, like if any new strain came, again if it is causing new diseases, again we have to collect the data, again we have to train these models. So only this training and getting the accuracy is the difficult part in this AI based disease detection especially. And another one is the rapid biosensors and diagnostics. Biosensors means the name itself indicating that here we are using two components, like biological component, enzyme, antibody and DNA, and another one is the transducer, which converts the signals, whatever we are getting, into the measurable output. Because until unless that signal can be measurable, we cannot able to detect the presence of the pathogen. And biosensors helps in detection of pathogen in minutes to hours, and we are having different type of biosensors like immunosensors. Immunosensors is nothing but based on antigen antibody reaction. Like suppose if you are taking the ELISA, enzyme linked immunosorbent assay, that is based on antigen and antibody reaction. And another one, DNA RNA biosensors, which are based on with very high specificity, like nucleic acid, like genome nature of the pathogen. And these are used for viral diseases and fungal identification. And another one is the electrochemical biosensors, which measures the electrical signals during some reactions, and which is we electrochemical biosensors used to be available for the portable, which we can carry to the these net house conditions. And field levels also we can use. And another one is the optical biosensor. These are the four different type of biosensor we are using. Optical biosensors means based on fluorescence signal or color image, like if you are using lateral flow devices which are based on color change. Like these are optical biosensors and which helps in easy detection. If we will take the rapid diagnostic techniques, we are having the enzyme linked immunosorbent assay. These are helpful for before going to the sowing. If you wanted to screen your seed material, whether that is the healthy or not, or whatever transplanting, before transplanting, whether the seedlings raised. If you want to check, then we are having the different diagnostic techniques like enzyme linked immunosorbent assay and polymerase chain reaction and loop mediated isothermal amplification and lateral flow devices. These I will explain in coming slides, like in another presentation. Then if you'll see the applications in the protected cultivation, which is early detection, like this AI, early detection, it will be useful, and monitoring of disease spread in the greenhouse, and screening the planting matter, and we will get the rapid results, high accuracy and on-site detection. And it will reduce unnecessary pesticide use, because there is no need to use the pesticide without knowing the whether pathogen is present or not. Only a limitation we are having, like if you'll take the sensor based devices, the initial cost of installment used to be very high, because for installing sensors and installing cameras to take the pictures, so these construction initial itself will be high, and need for skilled handling. If you want to screen the seedlings before transplanting, you should have the idea about this, how to screen the seedlings. And these are the some of the limitations. And another one, smart greenhouse and Internet of Things, is smart greenhouse means the greenhouse what we are using for protected cultivation. It used to be equipped with all the required things which are required for monitoring of the disease. Like it consists of sensors and Internet of Things plus automation system. Like cameras used to be fixed in regular positions, and they will take the images and they will send to this, with the help of sensors, they will send to the monitoring system. So it will helps the real-time monitoring of the disease diagnosis. And Internet of Things in agriculture means that network which is connected devices and sensors. These all things has to be connected, so that there used to be for each and every corner of the this protected area. It used to be screened with the help of these devices, and it will send the information to the monitoring system, whether any environmental changes are there in that protected cultivation, or whether any pathogen infection is there, so that with that information we can get that whether is there any chances for the disease development. So what are the key components? If you will see the smart disease diagnosis, like the sensors which will monitor environmental conditions, because monitoring environmental conditions is very important under glasshouse, these protected cultivations, like temperature, humidity, soil moisture and leaf wetness, and light intensity. And imaging devices, already I told, like cameras, like RGB thermal multispectral imaging, and capture plant images which will captures the regularly, and this is especially helpful for early symptom detections. Then data processing units, because whatever the cameras are capturing the image, they will send it to the processing units, like microcontrollers, and they will collect and process the sensor data. These are the softwares like Arduino and Raspberry Pi, which will, these are the microcontrollers, they will process the data whatever collected by these cameras. Then after this microcontroller, they will send the data, and they will store the data, and that is cloud and data storage, and which will we can access the data also, that is remote access is also possible. And like if you want to access the data from one location to another location, which will be possible because of these smart greenhouses. And also helps in AI based analysis. And we can screen this, we can observe this data either through mobile apps and dashboards also. And for we can give alerts to the farmers also, even under field applications also, it will be useful to give any suggestions to the farmers, any alert to the farmer to take like a warning system to the farmers. And how these sensors they will work means, first sensors will collect the environmental data, then captures will these cameras will capture the crop images, then the data will be sent to the cloud via Internet of Things. Then these artificial intelligence algorithms, they will analyze the disease symptoms and what are the risk conditions. Then system generates the alert that whether the possibility of disease is there or not. Then farmers receives the recommendation which fungicide they have to spray or whether any insecticide they have to spray, so that he will receive the recommendations. And how the disease prediction will happen through the Internet of Things means based on environmental threshold only. Like if high humidity is there, there is a possibility of fungal diseases, and based on leaf wetness, downy mildew risk used to be there. So it will predict that before symptoms appear, that is spread of the fungal diseases. Before spreading itself, it will give the warning system so that farmer can take the precautions. So if you will take some examples how the people managed powdery mildew management in greenhouse in case of cucumbers. This case study was from the Netherlands. So they found that they by predicting this high humidity which used to favor this powdery mildew which is caused by Podospira xanthi. So they used a resistant cultivars and they reduced the humidity under protected cultivation by improving the ventilation, and they use the biocontrol agent like Ampelomyces quisqualis and fungicide rotation like contact and systemic rotation used to be there. Because of this, they reduce the disease incidence by 70% and they also improve the yield. And another case study from the Israel, how they manage the tomato yellow leaf curl virus, like utilization of reflective mulches to reduce the whitefly landing, and insect netting on greenhouse vents and resistant hybrids, and regular sticky card monitorings. So because of using these classical approaches, there was a reduction in the whitefly population and tomato leaf curl virus incidence. So we will see some of the advanced detection techniques, what we generally use. Is it visible, this presentation? Anyone?
Yes, visible.
Yeah.
Yes, yes, ma'am. It is visible.
Yeah. This is detection methods is common for all the pathogens, whether it is a fungal, bacteria, viral, especially only few techniques will be different. So I will explain most commonly used techniques, generally what we used to follow. So these are the overview of the different techniques what we are having. So convention, these are seed detection methods. Yeah, this is the very important serology based assay, that is enzyme linked immunosorbent assay. This is the most used method for the virus detection and bacterial pathogens. So basic principle, and here we will use two different substrates like PNPP tablets and tetramethyl benzidine. And I will explain in detail. This is the ELISA plate where we can test around 96 samples in a single go. Like if you are having different seed lots, you from where you raised the different seedlings, so you can test 96 samples in a single go. And we are having a different type of ELISA, but generally we will use either indirect DAC ELISA, generally we will use, or DAS ELISA we will use. So here you can see DAC ELISA I will explain. If you will take the single well, like if you wanted to test any infected, suppose in a protected cultivation, if some plant is infected with tomato leaf curl disease, if you wanted to test that whether this is really infected, that plant is really infected or not, suppose 10 plants you collected, then you have to grind and you have to pour that supernatant into the ELISA plates. Then we will incubate overnight. Generally, we will prepare the coating solution, like it is phosphate buffer only, and we will add some solutions. So in that solution, we will grind the leaf sample. Then we will load 200 microliter of that leaf sample into the single well. If 10 samples we are having, then 2 replications, generally 2 to 3 replications we will follow. And after loading that supernatant, we will incubate the overnight. Then next day morning, what we will do, we will decant the solution, that overnight incubated solution, we will decant, because these ELISA plates which are having protein binding capacity, like a virus particle, which is nothing but it is a protein, because nucleic acid enclosed with the coat protein. So these pink color, what here we are showing, this is the virus particles. They used to absorb to the this ELISA well. So after decanting the solution, then we will, if we used to load the phosphate buffer, then we will incubate 3 minutes for each wash. Like after three, then we will wash 3 times to remove the any dust particles present in the this ELISA well. After 3 washes, then we will add the blocking solution. That blocking solution is nothing but milk powder, what we used to use in regular use. That milk powder, which is used to be dissolved in buffer. Then we will load and we will incubate at 30. All the ELISA steps we have to carry out at 37 degrees centigrade only, because if temperature deviation from 37, we will not get any proper results. So after blocking solution, we will incubate 1 hour at 37 degrees centigrade, and after blocking, again we will take out the plate and we will decant the blocking solution, because already your virus particles, if virus is there, then already it used to be absorbed to the microtiter, these ELISA plates. So after that, then we will add the specific antibody that which wire, if we want to detect the tomato leaf curl virus, then we should procure the same antibody which can capable of detecting this pathogen. Then we will dissolve this antibody in phosphate buffer saline PBS TPO solution, that is phosphate buffer saline with a tween 20, and we will add chicken albumin and PVP in that solution. So with that solution, we dissolve the primary antibody in that solution. Then we will add 200 microliter of primary antibody to these wells. Then again we will incubate 37 degrees centigrade for 1 hour. So 37 degrees centigrade, 1 hour is the standard for incubation. Then after 1 hour incubation, again we will decant the solution. Then so for this virus particle, this primary antibody is already binded. Then we will add the secondary antibody, which is universal conjugate. And again we will incubate for 37 degrees centigrade for 1 hour. After 1 hour, again we will decant the solution. Then finally we will add the substrate buffer along with the enzyme, so that this enzyme will react to the secondary antibody and it will give the color precipitation. So if you are getting the color precipitation like yellow color, because here already I told we are using two substrates like PNPP. If we are using PNPP, then we will get yellow color reaction, like here you can see. Another TMB if we are using, then we will get blue color reaction. So these are the different type of ELISA: direct ELISA, indirect ELISA, and the sandwich ELISA. These are the different type of ELISA we can follow. Generally, most of the laboratories used to follow this DAC ELISA or DAS ELISA. This then DAS ELISA, this is sandwich ELISA. So these are the two different techniques we will use. And another one is the PCR reaction. Another seedling grow-out test is also there. You can observe the seedlings which you raise it from the seed material. And another one is a polymerase chain reaction. This is the most useful technique for detection of any pathogens from the protected cultivations, where you can see, just to suppose some example, this is the infected leaf samples. Here you can see these symptoms and from seed material, like we collected the seed material from these leaf samples and we detected through the polymerase chain reaction by amplifying the particular region of the viral genome. Through this, we can confirm the whether pathogen is present or not. And different variants of PCR there. In these variants, only we need only real-time PCR, because the real-time PCR is the, I will show you. Yeah, generally cyber green dye based assay we will use. In real-time PCR, we can detect the small quantity of the pathogen also, because sometimes in PCR what will happen, if the pathogen used to be present in minute quantity, then we cannot able to detect that pathogen. But in case of real-time PCR, even if it is present in nanograms also, very less quantity also, we can quantify that pathogen. That is the advantage of real-time PCR. And this PCR, and another one is the reverse, this multiplex PCR. In the previous lecture I showed that detection of multiple pathogens. If you want to detect the multiple pathogens in a single reaction, then we have to standardize initially the primers for detection of different pathogens like tomato leaf curl, New Delhi virus. If we will take the example like Palampur virus, Joyadpur, we can detect in a single reaction by standardizing these primer conditions. Here you can see detection of onion yellow dwarf virus and LXZ virus simultaneously in the single PCR reaction. These are the 13 samples where the presence of both viruses we can detect in a single reaction. And another recently people are using that loop mediated isothermal amplification where most of the laboratories they don't have the PCR missions. In such cases, if we are having the hot water bath or dry bath which can maintain the temperatures, so we can go for the loop mediated isothermal amplification. Simply we have to mix all the PCR reactions together and we have to keep the water bath after standardization, whether 35 degrees or 40 degrees after standardization, then we can see the reaction. Here you can see the presence of pathogen in the 4 samples. Here we could not observe the any reaction, that is absent. From these samples pathogen is absent, and from these 4 samples pathogen is present. This is the loop mediated isothermal amplification. But only problem is that standardization used to be the difficult in this part. After standardization, it used to be very easy to use. And another one is the RT-PCR because we are having the DNA viruses and RNA viruses. Tomato leaf curl New Delhi virus is a DNA viruses. If you will take the cucumber mosaic virus and zucchini yellow mosaic virus, these are the RNA viruses. In such cases, first we have to isolate the total RNA from the leaf samples. Then we have to synthesize the cDNA with the help of reverse transcriptase. After synthesizing the cDNA, that is complementary DNA, then we can proceed through the polymerase chain reaction PCR, where we can observe the amplification of that particular pathogen. So if you wanted to improve the specificity, then we can use the ICRT-PCR, that is immunocapture reverse transcription polymerase chain reaction. Especially in case of low quantity of the pathogens, we can use this ICRT-PCR. These are the different methods we use for the detection of pathogens. So any doubts in these things?
Hello.
Hello.
Hello. If you have any questions, please ask. Hello. Am I audible?
Hello. Yes.
Yes, yes, yes. Yeah. Madam, I'm Dr. Jambamma, PJT.
Yes. Yes.
Madam, regarding that modeling, you told that in protected cultivations, which models are very popularly they are working on modeling?
Generally, up to now, only the CNN models everybody is using.
CNN.
CNN.
How much data points they are taking? I mean,
How
any
much?